Dr. Lorrin Pang is an anomaly. He's a public official. And most people that are in these positions, they do what their boss says. They just keep in the box and go along. Not Dr. Pang. 12 years ago, he was here in the Paia Community Center, and he first met GMO-free Maui. He didn't know anything about the issue before that day. He connected with them. He thought, oh my God, this is really important. He then went with them to the state convention, and in that time, they've done 4 court cases, and they've won all of them. He's also gone to New Mexico about 4 years ago. He's going to Arizona. He's gone to Thailand 9 years ago. He was in Brazil the last 6 months, and he spoke 4 times there. So we are very, very fortunate to have him here. Dr. Lorrin Pang.
I don't think this one. I've always asked to debate. I'll debate whoever wants to debate with us on the TV or not on the TV. But I do have to tell you a little bit about my background. I was born and raised in Honolulu. My grandmother, maybe my grandmother's mother, is from here in Paia. They came in as sugar planter workers, and eventually my grandfather became a banker. I guess they saw the promise in him. On my father's side, things were kind of rough. They started out as duck farmers in the headwaters of Paoa Valley, that's a little bit outside of Honolulu, and he died in the swine flu epidemic of 1918. So that left him with a stepfather who was, typical stepfather syndrome. So I inherited nothing from my stepfather, although he was a very, very smart guy. He wasn't in my lineage. I'm basically a duck farmer and a banker. I've given this talk almost on a yearly basis for the last 6 years in Kauai with a college. And usually from GMO-free Hawaii, there were 3 advisors, and then there's a member. There's one advisor for legal things, Paul Atichoff. He'll win all the suits. Then there is a ecologist, agriculture expert, Emeritus Professor Hector Valenzuela from the UH. He's fought with them. They threatened to cut off his funds, and he always protests, and he's very prominent. And then I will represent the health issues. So we have health, environmental, and ag, and legal. But as we go out to try to move the bill forward, you can imagine a room this size where maybe half the people don't know the issue, the other quarter really hate you, and a third, well, that doesn't add up, 10% are going to tell you you're affecting my job, okay? Then you have to kind of look in yourself, have the gumption to say what you believe is true, and hopefully I'll convince you some things that are true. I will tell you, this is basically the same presentation I've given for the last 10 years, but I keep adding things that the industry, Monsanto, Syngenta, Pioneer, they keep bringing up. And so you have to be able to think about them and respond. But let's get really basic here. The issue came about initially when we tried to move legislation because we're trying to protect taro or kalo. Kalo is our native plant. It's almost synonymous with the Hawaiian culture because the firstborn son of the goddess, the firstborn son died, and they buried him, and from that spot sprung the kalo plant. The second-born son, Haloa, is Haloa the man. So there's Haloa the kalo, Haloa the man. And they told the first man, if you take care of your brother, he will take care of you. And to modify or patent or mess around with Hawaiian's kalo or taro is absolute no-no. But we went and tried to, we blocked the genetic modification of kalo. The state would not hear of this, so we went county to county. So Big Island, they blocked it, and it was unanimous. The mayor overrode the veto. Then they overrode the mayor's veto. And at the very last minute, the coffee farmers said, yeah, we want GMO-free coffee too. So they kind of got on that. The next, within 9 months, we were here blocking GMO kalo in the field, in the market, and in the lab in Maui. Oh, by the way, the field, lab, market, it was blocked in the Big Island. Because when we saw GMO rice escape at the turn of the century, 2000, no one could figure out how it escaped, field, lab, or market. So if you can't figure that out, you block them all. Okay? So on Maui, we won, and it was blocked. Now, I have to bring this up now because it's kind of a warning. We were in the middle of giving testimony before our county council about the uncertainty and risk of GM kalo. I always speak about GM food, but the Hawaiians were there talking about kalo. Fine, we travel the same path sometimes. In the middle of the testimony, in the middle of my testimony, I told our county council this might be the last time we talk about this because we are about to be preempted. That is, the state pushed forth by the feds passed a rule that no county can be more conservative for GM crops. Now, that's a very strange preemption. Normally, the preemption is a county can be more conservative, but not more liberal. This one, it was twisted. A county can be more liberal, but not more conservative. The county members were furious. They do not want to be preempted. And so a lot of them kind of interrupted the meeting, pulled the law, read it, and started talking because they filmed the county. And we kind of won that one, not on the issue, but because they were so upset about being preempted. There's always attempt to preempt the genetic modification control. There always is an attempt. So people are watching this, and all that we do, lo and behold, one day they might say, it's illegal. You preempted. The other issue they might bring up one day, we're always afraid, is stop talking about this. This is a national security issue. No, wait. After 9/11, we had these talks for Homeland Security. And these guys came in from Texas, and they talked about food security. And this guy from Texas, he kind of looked like Dick Cheney, so I kind of treated him like Dick Cheney. He said, monocrops, GMOs, are the most dangerous thing of all. And I thought, hey, that's useful if I quote him. So after the conference, I went up kind of innocently and I said, can I quote you? He goes, yeah, sure. Monocropping, GMOs are very dangerous. When you get wiped out, you get wiped out. Now, I thought that was understood, but to hear him say that. So I said, well, so you'll speak against them here on the islands. He said, you don't understand. I said, what don't I understand? We want the enemy to have it, Iran and China, so you never have to invade them. You just say, I'll turn off your seed, or your seed might time bomb in 3 years. We want them to have it, but not us. And so I said, well, then why do we have it? I mean, we have it because we're sending them the seed. He said, well, you kind of got to show that we have to have it so that they can think it's good for them to have it. It's like, whoa, I have like no idea. And that's when I decided, forget the politics, just do the science as I was raised. I went to high school here. I went to Princeton University. After 4 years of mathematics, physics, and chemistry, I graduated with honors in chemistry. I did my medical school in Tulane in New Orleans. I have a master's in tropical medicine. After that, I spent 24 years overseas with the Walter Reed Institute of Research and the World Health Organization. My job was to test drugs, vaccines, and diagnostics against tropical diseases. In the year 2000, after 24 years overseas, we decided to come back because my parents were old, and I came back and I saw this problem here. So let's just get going here. Okay, the title of this talk, and it's the same title in Arizona, GMOs, genetically modified organisms, are we gambling with our future? And it's a trick question because the answer definitely is no. And people say, wait, stop, whose side are you on? I can guarantee you, as you define gambling, we are not gambling with our future. Okay, here we go. And this is how I define gambling. You know, you play roulette. When you play roulette, you know how many black squares, you know how many red squares, and there's 2 greens. So you know the chance of losing, you know the chance of winning. And the chance of losing is always a little bit greater because there's the equal number of black and 2 greens. Okay? But you know the odds of winning and losing. Okay, so that's how I define gambling. Let's talk basically what is genetic modification. Now, genetic engineering, it's almost, they use the terms interchangeably, GE, genetic engineering, or GMO, genetically modified organism. So they try to engineer. This is the ideal situation This is what they want to do, their intention. So basically, they take the host, let's say it's a tomato. The tomato has a genome. The genome is the sum of all the genes linked together. For example, humans have 33,000 genes, and it's forever increasing because some of those pieces were called junk DNA, and we said, well, those aren't genes. And people said, yes, they are genes. But very interesting. Every day we learn that those junk DNA, genes or not, are sometimes more important than the gene. But all the genes strung together are your genome, and that's your DNA. So all you really want to do is take the gene from a foreign organism, like the fish, and you stick it in the gene of a tomato. You're going to stick the thing in. Here it is. It's stuck in. And the gene is read by the RNA. I know that's hard to read, but it's read and makes a protein. The protein is what makes you, you. The DNA is the blueprint, but the protein expresses the gene. And so, for example, I can't remember, but blue eyes or something. There's what, 6 genes that control that. The length of your eyelash. There's a gene signals that fix that. I mean, how come your eyelashes don't grow this long? I mean, normal people. Okay, oh, that's your eyebrow, sorry. All right, so that's how it's called. The gene is expressed in the protein. But what's this blue and what's that green thing? Well, if you're going to put the gene in, you got to turn it on. 98% of our 33,000 genes are quiet at any one time. So some are on when you're 3 years old, only when you're 3. Some are on when you're 75, only when you're 75. And God help you if you get the signal switch. So sometimes we see little kids, they prematurely age because they turned their gene on at the wrong time. Some genes, onco is cancer. You have the gene to make leukemia cancerous. And not just one of them, there's probably 10 leukemia genes. If you turn those on, you get leukemia, okay? So they're silent, and it's orchestrated turning on at the right time. But if you're gonna put the red gene from the fish into the tomato, for all that trouble, you wanna turn it on. That's the green thing. It's called the promoter. The promoter is like an on switch, okay? Then there's a blue piece called the marker. When you put this thing in, you wanna know if it got there. Well, what's the problem? Because the way you put it in, how do you actually put this thing in? You get a petri dish of all this DNA material from the tomato. Then you get your little construct, the promoter, the marker, and the gene. You coat metal, you put it in a .22 shotgun thing, and you blast it into this plate, this receptive plate of cells with the DNA. And if it goes in, then the whole thing is turned on, and it starts to make the protein marker that I didn't draw. The protein marker is how you screen it to say, we got it. We got success. If it doesn't get in, you don't make the marker. And so that's how you blast away with the marker. And that's how it's supposed to work. That's the ideal situation. But how does it work? When you take the construct and you blast it away, it is true, sometimes there's a virus that can carry it. It's either virus or shotgun. It's kind of same effect. Look at this. You blast it away, and look what happened to your construct. Hey, there's a free promoter. Hey, there's a free marker. Oh, that thing's in the wrong place. Oh, that thing is sitting on a gene or broke the gene. What's going on? Look at this. Here's the other one next to it because you blasted in a plate full of dishes, a plate full of cells. Look, you see those promoters? By convention, just they turn on, they're on switches, turning on the things downstream or to the right. I made it turn on this little blue thing. That blue protein is supposed to be silent. Maybe that's your oncogene, cancer. Maybe that's supposed to turn on when you're 75 or 3. Whatever, you turned it on. Do you know what you turned on? Well, look, here's a successful piece right there. Isn't that good enough? Yeah, that was good, but what about all this other weird stuff? Look at that, there's a free gene here bridging the gap with no promoter. I guess that's silent. It's a mess, okay? Furthermore, I was wrong to draw the genome like a line. It really is a three-dimensional twisted turn. I do it like a mumbo jumbo, but it is quite organized. It's called flip in, flip out, writhe, torsions. And why is that important? Because when it folds on itself and you try to read the frame, sometimes you read cross-bridge. It's called the shifting frame. And so if there's one thing we learned in the last 10 years, it's how little we know about genetics and genome. Now, the scientists will tell you, I'm a scientist, but you should know how little you know, and that has been shown in the last 10 years. When I went through school, the idea was one gene makes one protein. If you put anything else, you got an F. That was so basic, you got an F. Now we know in the humans, we have 33,000 genes. It makes 100,000 proteins. So on the average, how can one gene make 3 proteins? I thought you said it reads it down the line. Well, it reads it crosswise. The frame can shift. It can read this part and continue on. Now that's stupendous. We went from one gene, 1 protein to one gene, 3 proteins. Furthermore, there's tons of material in between that we had no idea what we were doing, and we called it junk. It turns out to be the orchestrators. It's called the RNA series. Never mind if you can't read it, okay? That's one thing. One gene doesn't make one protein. The next thing we learn is a lot of our genes are shared amongst the plant and animal kingdom. Whoa, you mean my area is malaria. The malaria parasite shares a gene that makes a pump that pumps out drugs. So every time you try to poison the malaria parasite, it's resistant because the drug pumps it out. Guess what? That pump, that gene for that pump is in my liver. That's how my liver detoxifies things. How in the world did I get the same gene as the malaria parasite? Do you think we had common ancestors that had sex? No. That's a tantalizing thought. What really is true is that there are gene exchanges through the viruses. The viruses go into an animal. It hijacks and it incorporates itself in the genome, takes it over, then moves out and infects another species. And it will share the genes. If that gene is useful, like this pump to detoxify your liver, to detoxify the malaria parasite, if it's useful, we can retain it. If it's not useful, and our ancestors have paid the price, in history of mankind, I guarantee you, viruses brought us some pretty weird, useless stuff. Well, that lineage of people died out. We have paid the price. Remember that. You are who you are, kind of adapted to this environment because your ancestors paid the price. Okay? Now, one more interesting thing we always learned. So some genes are silent and they shouldn't be turned on. We share a lot of genes, and then there's this one gene making 3 proteins. Those silent areas, they used to call junk DNA. We are learning that they're the orchestrators. And I want you to remember, this is very good. Sometimes the analogy is kind of infantile, but you can remember it best this way. You have a gene from the fish. You put it in the tomato, okay? That's like taking the tuba. I was in the band, you know, the high school band. That's like taking the tuba player. I didn't play the tuba. I played the trombone. That's like taking the tuba player and throwing him in the football team in the middle of the game. First of all, the tuba player, when he lands in the football team, I guess he knocked out the quarterback. Okay, fine. Can't help that. Next, the tuba player, the on switch promoter is always on. So he's playing and you tell him, shut up, we're trying to play the game. But he's always on and there's no orchestration, okay? And the final thing is right here, right here. These genes that don't belong there, they're from other species, they're called promiscuous. They either move out because the tuba player doesn't belong in the football team and they push him to the girls' volleyball team and he's happily playing the tuba, being disruptive. These are called promiscuous genes. Genes that don't belong there move out or they amplify. In other words, one copy, oh, there's one. The next time it replicates is 2, 4, 8, 16. So amplification, amplification. That was my area of research. There was one poor worm, we gave him a foreign gene, and after 7 generations, I think 56% of his gene was that gene. What do you think he's doing, making tons of useless protein? This has to do with GMO crops, doesn't it? You put in a construct that forever is producing that red thing. You're not supposed to turn it on all the time. You're supposed to turn it on when you need it. That's called epigenetics. So you made it so it's always on. That's wasteful. And when you do wasteful stuff, you are attacked by the other microbes, the viruses, the parasites, the fungus. You forever weaken the other areas. That's like a guy who's so obsessed with football that he can't cross the street. That is the why GMOs are almost synonymous with sterilizing the fields so that they cannot be attacked by the other microbes. Now, we learned this, and they learned this too, because it wasn't supposed to be like that. But that's why the suit is in Kauai. The amount of pesticides they use. You think sugarcane, you think pineapple's bad. You should see what they're doing in the GM fields. We sued, there was disclosure, and some of these pesticides, I have no idea what they are, but there's a whole bunch of them, okay? So remember that. Because of the nature of the setup, that's how it's gonna go. These are the references that tell you about these moving to other species. That's called horizontal gene transfer. Okay, so that's basically what happens, and that was the intention. Now, look, everything I told you was plants, plants. So they take the plant, they take this modification, there's a mutation, they want to see if it's stable. You put it in, but all kinds of weird stuff might happen. So you bring it out to the conditions, and you put it out here under rain, sun, fertilizer, just to see if it's good, if it's stable, consistent, okay? Then all the weird stuff dies, and you amplify the good stuff, and you select the consistent product, and then you make seed and ship it out for other people to grow, and you say, this is probably consistent. So all the plants are plants. Plants are plants. And so the industry, they always say, Dr. Pang, you're a human health guy. Don't talk about plants. Plants are plants, and humans are humans. Okay? In the humans, let me press the button here. In the humans, we have done GMOs long before the plant guys did it. And we called it gene therapy. And this was not taking tomato to human or fish to human. It was taking human to human. Hey, I'm matching the species. What more do you want? And so don't try to read this. This is just a quote because guys who get the PowerPoint can read this themselves. There were two boys in Philadelphia. They had severe combined immunodeficiency, SCID, boys in the bubble. They couldn't come out. They would be attacked. They didn't have immunity. And they got real tired of living that way. And so one day, the medical people said, would you like to try to take the gene that's in everybody else and put it in you so you can live like normal people? And they said yes, okay? Now, when you put the gene in the boy that gave them immunity, you stuck on a promoter. Like I said, why put it in if it's going to be quiet? Okay, and they might have put on a marker, I can't remember. So they injected the boys, and the genes went into the bodies of the immune cells. And what happened? Did they get immune? Well, we don't know. They died of cancer. You turned on the leukemia genes by mistake. This is published in New England Journal of Medicine. This is our lead medical journal. Furthermore, they did a study to say, whoa, how unlucky. You turned on the wrong gene, the leukemia gene. That was more than luck. They actually calculated there was a 4-fold above luck. In other words, those leukemia genes called the promoters. They can't wait to turn on. And so the medical people, we have a moratorium on gene therapy. Gene therapy has been the biggest disappointment ever in the last 40 years of medicine. It was so much promise, but when we realized the genes are so complicated that we have a moratorium until you can put exactly where you want it, not turn on the leukemia gene. You can turn on exactly what you want. So even if you put in the wrong thing, don't turn it on. If it's a mistake, you can pull it out or you can turn it off. The 4 conditions. We can't do one of them. And so the medical guys, we're very alarmed and we don't do it, okay? But the plant guys, hey, well, the plant guys are plant guys, except for one problem here. Now, let me get this. Here's the problem. Here's the problem. When you grow these plants and eat these plants, these doctors, Travik from Norway, Ermakova from Russia, Putztai, Pakistani in England. Putztai's work has been questioned. Even he admits something wasn't quite right. But Ermakova, Travik, there's tons of them now. They have shown that an animal, like a mouse, when fed GM food, the construct, the marker, the promoter, and the gene will go into the stomach cells of the rat and turn on within 2 hours of first feeding. That is one heck of a transfer. But I warned you that these are promiscuous genes and they go out and turn in. You've constructed the gene so it can cross species. Well, what did you expect it to do when you go into the mouse? And it not in the mouse stomach, in the mouse stomach cells, and was on. Then in 6 hours, the stomach, the blood goes up into the lymph nodes. It's in the lymph nodes and on. So that was the mouse or the rat, maybe the rat. But they also, Travik did cultures, human tissue cultures, kidney, intestine, not a human, but he puts these genetic things from the plants on and they incorporate in the cell tissue. The stuff is crossing and it's active. Even Monsanto will admit they're active, okay? So the stuff does cross into mammalian and human cell lines. Dr. Ermakova, she was Russian and she did very interesting studies. She fed the mice GM food, and the mice she was looking at birth defects and diminished litter size, which means birth defect in utero, so they never delivered. And she saw a bunch of them. The industry tried to repeat it and couldn't repeat what she saw. She tried to repeat it and couldn't repeat what she saw. She tried to repeat it again and saw it again. The stuff, sometimes you see it, sometimes you don't. Sometimes you see it, sometimes you don't. In my field of science, drugs and vaccines, you will not do any study in the lab, in the animals or in humans, until you can produce the product consistently. There's a principle called GMP. It's almost like GMO. Good manufacturing practice. If you cannot produce it consistently, don't bother testing, because the testing will be all over the board, which seems to be what we see. But that was perfect for the industry, because when Ermakova showed it, and they didn't, and then she didn't, and then they will use the negative data, looks safe to me, to negate her positive data. That's not how science works. You don't use negative data to refute positive data. You want to do that? I can take, all of us decide we should smoke. Some of us get cancer, some of us don't. The guys who don't, does that mean these guys don't get cancer? You don't use negative data to refute positive data, number one. Number two, you don't even try to interpret a study until the product is made consistently, okay?